All right, good morning. Before we start, I just wanted to make two quick announcements. One is, if you're scheduled to go to the nuclear reactor, you would need a photo ID. It's a secured facility, obviously. The second thing is, the voting booth is open. In case you need to leave early or have already made up your mind, the voting booth is down here. I think the majority of you will probably vote after the next poster session, but just to let you know in case you need to leave. And I now turn the mic over to Squire to introduce the speaker.
Good morning again. I am super excited to welcome Professor Patricia Babbitt from University of California at San Francisco and her research scientist, Dr. Shoshana Brown, as well. Dr. Babbitt is a leader in bioinformatics, especially in using bioinformatics to take tertiary structures of protein and sort of figure out what their functions might be. And so as we now move into this post-genomic area where we have this huge amount of sequence information, we can now take that information and look for relationships between or among enzymes that you might be interested in and other proteins that are out there. And this work is also really important in our ability to sort of annotate what's out there. There's a large amount of sequence information out there that we have no idea what the protein does that the actual sequence encodes. And so this work is becoming just more and more important. I mean, we can learn about all of these spectroscopic techniques and all of these other techniques to look at enzyme structure or cofactor structure and function. But nowadays you really need to look in terms of reactivity and groups of enzymes that might bear, that type of reactivity. And so using Dr. Babbitt's methods, sequence similarity networks, that can enable you to figure out, these interrelationships between, certain proteins and others that are in the database. We've come to this era now, at least here in the United States, where you can no longer really get funded to study one protein in isolation. You need to understand that protein's reactivity and how it relates to all kinds of other proteins that are out there. And so without further ado, Patsy, welcome again. Now today she's going to give a lecture that's a little bit more advanced than she gave 2 years ago. 2 years ago it was very general and basic. That lecture is recorded, and you may want to go back and look at that. Okay? Thank you.
Great. Can you hear me okay? So I am going to talk just about protein similarity networks today. Bioinformatics is a huge, huge topic in actually many different fields at this point in time. And I'm really going to focus in on similarity networks and how they help us understand something about structure-function relationships in proteins. The focus of my lab has been on enzymes, and because they do chemistry, we're looking for very explicit mappings between sequence and structural features and functional features. And we're doing that in a large-scale context to give us some broad perspective on that problem. What I'd like to do today is, instead of just talking at you for 90 minutes, I'm happy to answer questions. I think this is a very complicated area, and it's a new one. And so you may have some basic questions. We'll try to keep a balance so I can get through most of the material, but please feel welcome to ask a question. I'm really going to focus, I'm going to give you some just basic background to get everybody oriented. Many of you may know quite a lot more about this than others, so I hope we hit a happy medium. I'm going to talk about how we use protein similarity networks along with many other methods. They're never an answer by themselves. And I think of them as a hypothesis machine. It doesn't give us the answer at the end of the day. And then I'm going to talk some about challenges for interpretation. It turns out that these are very, very complicated representations, and many things are going on. There's a lot of heterogeneity in the similarities you capture in the pretty pictures we see. I'm going to show you pretty pictures today. These are actually, networks are generated computationally, and you can study them analytically. But there's an enormous amount of value you can get in terms of biological and chemical intuition from visualization of those pretty pictures. But knowing a little bit more about what's behind them may be helpful for you if you want to use them. The thing that really drove us to get started in this area is that we've been studying enzyme superfamilies for a couple of decades, essentially my whole career. And as we've gotten more and more data, these superfamilies have gotten much bigger. So now we're working with sets of proteins that are sometimes well over 100,000 sequences. And we don't have adequate ways to handle those in terms of really probing the structure-function relationships. To really look at a multiple sequence alignment, you get much bigger than 100 sequences, and you really can't do very much with it in terms of trying to make inferences. It's just too big. High-quality phylogenetic trees, which is the standard in the field for looking at relationships between sequence proteins, is really limited. You have to get a really good alignment to get a really good tree. We now are using methods that come out of computer science and Tandy Warnow at University of Illinois that allow us to align thousands, sometimes tens of thousands of sequences. But they're very specialized methods set up to give us a mathematically optimal answer, but that doesn't mean it's a biologically useful one. This is the basic slide I use to explain networks kind of as a first pass. All of you have seen something like a phylogenetic tree or a dendrogram where you may have three different functions, blue, green, and yellow. And if you build some kind of tree for it, what you hope happens is that the way the differences in comparing those sequences partition, you hope they track with those functions. So that if you have an unknown in here and it tracks with the green guys, you have at least the beginning of a hypothesis about functional properties of that unknown. This doesn't scale to the kinds of size of the sets we have now, and we really want to use as many of those sequences as we can because the context that gives us, we're starting to learn a lot of new things we just couldn't see before on a smaller scale. So instead, we use graphical network models. Here, nodes, which are the circles, represent sequences. They could represent structures. We make reaction similarity networks. We make ligand similarity networks. The nodes are whatever the object is that you want to compare. The edges between the nodes are the connections between sequences, which in our case, we're using thresholded networks. So we define a statistical significance score better than which you have to have that statistical significance to draw an edge. And I'll show you in a minute how powerful that is in exploring a very wide range of diversity in the sequence sets. In its simplest form, the networks are generated by all blast. We do that routinely on a large scale for multiple superfamilies, and you have to use something simple like that because it takes, to do 100,000 sequences, we actually have to compress the data and we generate the blast on 100,000 sequences, then compress the results to be able to view them. That's a big computation. It takes time. It takes careful development of the code to do that. So we like to use something that's fast. You could also use much more complicated metrics. We use structure similarity. We look at similarity now between hidden Markov model profiles, for example. So there are lots of different ways to do this. The bottom line that I want to draw your attention to here is that if you use a very permissive significance threshold to draw edges, that is, you don't require very high statistical significance, you get a hairball. All of these proteins are related, but you can't really see. If you didn't have the colors, you wouldn't be able to distinguish very much. As we make that threshold more stringent and require more statistical significance in the pairwise scores to draw an edge, the network starts to come apart. At the end of the day, you hope you find a sweet spot where you get this. That in reality doesn't happen very often because biology is messy. And as I'll show you in a number of cases, why functional boundaries don't necessarily track with sequence similarity. But it's a pretty good starting proxy. And that's sort of where we start from. Yes?
Ask a quick question. Can you say a little bit more about the reaction networks and the ligand networks as they relate to the protein similarity networks?
Yeah, so I didn't put slides in about that because it's another sort of specialized topic. But we're very interested in whether the topology of the network, that is how it clusters with respect to ligands or with respect to reactions, how well it matches and correlates to the protein network.
I see. So you sort of start off with the protein network, and as you start to annotate or know what things do, you can then see how they
Right,
cluster.
And we're actually building some, it's not really software, it's some code, so that we can compare those directly. So we like to capture EC numbers. We could take reactions and actually compare them based on just the transformation from substrate to product and quantitatively compare what those are, cluster those reactions, and then ask what do those clusters look like relative to the protein clusters. So that's important to know because for the sequences or structures, you'll get some topology that tells you something about how the members of a big set relate to each other. If you get a similar topology for the reactions or the ligands, then you know that if you have an unknown in one of those protein clusters, your ligand might be similar too. Interestingly, in many of the superfamilies that we've looked at, and radical SAM is a really good example, they don't cluster, they don't match at all. And that's sort of a negative result, but what it tells you is the problem's harder than you thought it was going to be, and you shouldn't make the easy assumption that just because the ligand of protein A, if protein A and B look a lot alike, doesn't mean the ligands of protein A and B are going to be a lot alike. And that's the bottom line.
So you're working on bringing those together?
Yes. But it's sort of a big implementation project, and I think we'll make a good foundational pass at it in the next couple of years. It's a hard problem. There isn't anything like it right now. Just briefly, this is as I mentioned, we can handle thousands of sequences. They're fast to generate. We use Cytoscape, which any of you who are taking the workshops will be working with today to visualize the networks. And I guess I said a lot of this other stuff already, so let me go forward. We've done some initial validation to look at the use of BLAST versus the Smith-Waterman algorithm, multiple alignments, and phylogenetic tree distances, and we get pretty good correlation. That doesn't mean you can dispense with a phylogenetic tree. There's no evolutionary model built into these networks, and you're only using pairwise comparisons, which is the least rigorous way to get an alignment. So there are lots of issues, but we get something that comes close enough that it gives us a ballpark idea of how very diverse sets of proteins might relate to each other. And we've never seen that before until we look at this scale. So it's a tool that has its limitations, but it also is very powerful for getting you started in thinking in some new directions. Here's a network of the entire radical SAM superfamily. And what you see here is we've just colored different colors, major groups that have some similarities to each other and that have names. So if we talk about this as the B12 binding domain, in our database we call it the B12-like binding domain, because they're not all B12-related enzymes, but they're more related to each other. The green guys are more related to each other than they are to anything else. It tells you something about where the spasm twitch-like large group of enzymes falls in relation to some of the other subgroups. So already you begin to see a little bit of useful information. The squares are the structures we had as of a couple of years ago. We have quite a few more structures now, but it's a very, very poor coverage of structure space relative to the number of sequences. So we know very little about it at this point. We want to look at trees when we can to compare them to the networks, and I want to spend just a minute on this slide. This is an old slide from the enolase superfamily that was made a number of years ago. Here's a careful phylogenetic tree of the mucinate lactonizing enzyme subgroup that Margie Glasner generated when she was a postdoc in my lab. She's now associate professor at Texas A&M, so it tells you it was a while ago. You can see that the red guys, which represent the O6-benzoate synthases, there's a very large number of them, and they're very diverse, which is why you have so many leaves in the tree. The mucinate lactonizing enzymes are quite similar to each other, so they fall into one branch in the tree. And then we have the dipeptide epimerases, which are pretty much all we know now, dipeptide epimerases that are in the gray and green, and a bunch of unknowns, some of which John Gerlt and others have made some headway on. This is the corresponding network. You see the red guys, all the clusters that are separated from the main cluster are separated because their similarity to any proteins in this main cluster is not as good enough to draw an edge at this e-value threshold. And we use e-values from BLAST as a score. We also use bit scores, but this is a little bit more easily interpretable by biologists who don't work a lot with the guts of the bioinformatics. You see there are a number of groups of OSBSs, which tracks quite well with the tree. What you see that is different from what the tree tells you is that you have this big region in the middle that if I wanted to align these proteins with these proteins, it would be very useful to include these ones in this sort of central hub. And that's the kind of observation that has become very important to our work, is to identify what the linkers are between two groups that might be very different, even though they might be mechanistically similar. You want to know what the sort of structural differences or transitions might be, and I'm going to talk about that more later. You also see that there are quite a lot. Everything in gray was at the time when we made this was unknown, meaning it hadn't been experimentally characterized and we weren't sure of the function. Now I would say essentially all of these proteins will do the mucinate lactonizing enzyme or some fairly close variant of it. And you can see there are a lot of them, whereas you only have one leaf here. So you see different kinds of things from the networks. They're best used together. Sometimes when you're working with very divergent sets of proteins, you can't build a tree. So this is what you get. And so you have to be careful, again, about the interpretation, because we haven't seen these before. We don't always know how to evaluate them. We also run into a problem of simply, even though the networks are great for looking at very large sets, they have limits. So if you want to actually look at a network in Cytoscape, you really can't work with a network on a pretty powerful laptop or desktop machine if you have more than 500,000 edges. We have up to 1 billion edges if we do the full network for radical SAM. So you can't even open that even on a very big computer. And so we have a way of compressing that, and the way we do it, and I'm going to mention this to you because it's important, because you're going to see a lot of what we call these representative networks or RepNets. We take all of the sequences and bin them by some percent identity value. So at 90% ID, all of the sequences put in one node have better than 90% identity, pairwise identity at every other node in the set. And that removes redundance. Sometimes you'll have hundreds of sequences that are very similar, so it cleans it up. Sometimes we have to go down to 40%, which is about the lower limit we're happy with, well, reasonably happy with, to be able to align the sequences within that node. So we have some handle on, if we wanted to look at all the sequences in the node, we'd be able to align them. You start getting below 40% and it starts getting sketchy. We then compute all the pairwise similarities between every sequence in the whole network, and then take the median or the mean or whatever we want, the highest scoring pair, the lowest, whatever works for a question, and use that to define drawing an edge. And so we're not just picking one sequence out of a big group. We're actually using all the information. There's still, doesn't give you the detail, but it gets you started. And I just wanted to mention that because I'm mostly going to show you representative sequence similarity networks. The key thing to remember is that in most of the superfamilies we're worked on, we're using a 40% ID cutoff, which is already distant enough that you may have multiple functions inside that node. And you can have 1 to 1,000 or more sequences in a single node. So when you see these pictures, they're very much bigger than what you might, if you were going to look at all the sequences, which we can't because of the limits of visualization. And I'll show you a comparison a little bit later. A good source for generating your own networks was developed by the Enzyme Function Initiative. Here's the link to that. We also generate networks, but we can't do it on a large scale, so we principally do it for projects in our own lab and those that we are collaborating with experimental scientists. Okay, so how to use protein similarity networks depends entirely on your question. There are a million different ways to put these together. And so if you go to the workshop today or tomorrow, you get a taste of some ways to do it, and I'm going to show you a few examples here. But you could pretty much, if you could ask a question that takes advantage especially of a large-scale context, there's probably a way to do that with the similarity networks. And it's this large-scale context that we're really learning a lot from, and I'll try to point that out as I go along in my examples. So one of those uses might be selecting targets for biochemical characterization. Kathy Drennan has been working on the SPAS structures for especially spasm domain containing radical SAM enzymes. And having the network and seeing where the structures cover and where they don't, you begin to get an idea of maybe which targets you might want to go after to fill out that space a little further. Obviously, they're important for functional clues about knowns and unknowns, and a number of other important aspects that I will talk about a little bit later. And then at the end of the talk, I'm going to talk about some special topics that I thought people would be interested in. So here's a network of the entire thyroidoxin fold class of enzymes that we did and published in 2009. And there are something on the order of 30,000 sequences that we used a redundance filter to get down to about 5,000. So there are about 5,000 nodes on this picture. Sorry, the resolution's not so great. It's partly the projector and partly this is a big picture. And some of you are way in the back, so you're probably seeing colored blobs. But we built this at an e-value threshold of 1 × 10⁻12. And what that meant was we get a little bit of skew when we use the e-value, so the reality is that that's not all that statistically significant. We have a few red edges, and those are the ones that we didn't feel that confident of in sequence. But we get a general view of the relationship of these sets of proteins. So these are the thyrodoxins, these are the peroxyredoxins and other redoxins, and then these are the glutathione transferases. You have a few outliers. These are the disulfide binding proteins. Here's the structure similarity network. So the glutathione transferases have quite good structure coverage, and you can see that by the big nodes in the networks, those of you who are close enough. And if we just take those structures and do the same all-by-all comparison, you could see that the thyrodoxins and the peroxyredoxins are the closest neighbors to each other. You could see the glutathione transferases are outliers, and you get quite a lot of similarity in the topology. The reason you can't use structure similarity networks exclusively, however, is that you may not have anywhere close enough structures to give you an adequate view of the whole space. This is sort of a hierarchy that we use to try to classify different levels of functional and structural similarity in enzyme superfamilies. And the reason I'm showing it to you is that you can use protein similarity networks to try to learn something about a whole superfamily. You can divide and conquer and look at smaller subgroups that are simply more tractable. And ultimately, you may want to ask questions about what are the specificity determinants that distinguish one reaction family from another within the superfamily. All those proteins look alike. They're members of the same superfamily, but you want to know what makes the dipeptide epimerases different from the OSBSs, for example. And that's and I'm going to show you some examples at each of those levels of granularity for how we use the networks. So here's a glutathione transferase superfamily. This is the entire network, thresholded at 1 × 10 to the minus 13. We have about 1,500 nodes in this picture. They represent 13,000 representative sequences binned at that 50% ID cutoff. This is a few years ago. Now this is a much bigger superfamily. The mean edge value for these edges is about 4 × 10 to the minus 25. And the alignment length is pretty close to the full length of the GSTs, which you want to see because you want to get a big enough part of the signal that you have some confidence that you're picking up pretty good relationships in those pairwise comparisons. So these are mapped to colors from the SwissProt database, which simply took the literature from 100 years of GST studies by several hundred investigators and put them into classes based on, they basically fall under similarity. Those are not functions. They're just groupings where someone would find in humans what was called the alpha class GST, and then others would find additional GSTs that looked a little different, were fairly, somewhat distantly related, and give them another class. So this does kind of track with similarity. There are less than 1% of the GSTs, which are heavily, heavily studied and very important physiologically, less than 1% have known physiological reactions. And that's kind of a stunning observation. And you can see that here because most of this is gray. And that means it's uncharacterized. It tells us a little bit about how very little we know. And I think this was one of the surprises for us. We knew that we didn't know what all the functions were, but when we started to look at these, we've never seen a superfamily where we know a lot. Most of them look something like this. And that means that we may have studied these proteins greatly, or these are the ones that are typically studied in humans, the alpha, mu, pi, and sigma. And these are mostly bacterial and include proteins that are involved in insecticide resistance, all kinds of specialized reactions. And we just haven't even sampled most of this space in any way experimentally. If I make the E-value more stringent, 1 × 10 to the minus 24, we take it apart. the network. And you can see now that there are quite large groups that have no characterized functions at all. How did we choose that threshold? Because that's a key question. In this case, we were trying to propose a classification system for GSTs that pretty well followed what the literature had done for the last 100 years and gave us a fairly even representation. And it was chosen specifically to help that community in thinking about target selection. We have developed some sort of additional plots to give users and ourselves a little better idea of where to start in choosing thresholds. And we've been working for a long time at trying to develop an automated method to choose good thresholds. I have a graduate student right now who's trying to develop a method to estimate functional boundaries as well as possible. It's a very, very hard problem. But what you could start with is you could look at the e-value cutoff where this becomes more similar. These are negative log 10 of the e-value. And then you can look at the length of the alignments. You'd like the longest alignment that gets you that you can get. And clearly here, this is full length alignment. You'd like to be up here. And the e-value that you can the least stringent e-value is sometimes around 10 to the minus 20. And so that tells you how diverse you can get and still get most of the alignment. That's a good rule of thumb for starting to identify. We also use approaches such as if we have complex domain structures, we divide the set into groups with similar domain structures because that's a good starting point. Sometimes we simply map what we know functionally and structurally on the network and then try to identify if we know that two functional groups and their structures are quite different, we try to then toggle our thresholds to that. It's really at this point an exploratory effort. We really want to move toward higher throughput and automated approaches. Because of the uniqueness of every superfamily, I don't think that's really going to happen very satisfactorily. We still got to do the thinking. We still got to do the experiments to test our hypotheses. And so this is not like you push a button, you get an answer out, and you're done. Doesn't work like that at all. Then we use biology. So one of the things we know about glutathione transferases is that they're highly promiscuous. And this plot tells you different kinds of reaction types that are known in each of these little groups. And you can see there's a lot of variation here. So we decided as part of the, when I was working with the Enzyme Function Initiative, that we would try to explore this on a fairly large scale. And we decided to work on the disulfide bond reductase proteins, which are shown in green here. This was a collaboration largely with Richard Armstrong, and he had been working on these proteins for some years. And we thought, well, that would be an interesting one to look at because it's sort of an outlier reaction relative to the canonical GSTs. And so here's just a blow up of that little group here. Oops, going backwards. Of this new subgroup. And here's the representative network that was in the slide a minute ago. And so then what you do is you make a full network of that smaller group so that you can now use all the sequences, and that gives you a better handle on the details. And you have the opportunity, if you want to express or get a structure, you can see, you can work with the whole set instead of a compressed representative set. So this is the set that we then wanted to work with. This is the full network, thresholded at 1 times 10 to the minus 31. There's 1,600 sequences in this set. In this set, we have only 130 representative nodes. So you can see there's a big scale difference. What you see here is the results of those studies where, to everyone's quite great surprise, all of the subgroups that have a green node showed disulfide bond reductase activity, which was just really startling. It was when Richard first discovered this, he thought it was a very specialized group that was just found in this particular similarity group. And I think this was quite astounding to see all of the different groups. And these were only the ones that we sampled. We're able to sample. We were able to express the proteins. Steve Almo got, I think it was about 27 new structures to corroborate what this is on. And this was then the foundation for the next step, where we wanted to look in more detail about how these proteins did the disulfide bond reductase activity, and a couple of others of interest that we chose for, in part because we had experimental acumen to work with them. So this is the result of that specificity question. And it turns out that from the new-like subgroup, which is the one Richard had originally worked on, oops, I'm going backwards, sorry. That's this subgroup versus this main 3 omega subgroup and the Z-like subgroup. You can see that there are 3 different structural strategies for supporting that reaction. That was another surprise to us, and we do see this all the time in these big superfamilies, where we'll see the same function invented more than once in the background of the superfamily. So you can't even make an assumption that the mechanism for a particular reaction is going to be the same throughout a whole set of proteins that are of the same fold class and use fundamentally the same machinery to do the reaction. Was there a question? Any questions so far?
Nancy, for example, what's the function of the threonine and the cysteine then?
It's like how, when you don't have, when the column doesn't match all the way down, yeah, we've got cysteines, but then what are the T's?
We don't really know. Richard was working on it. The thing that is striking here is that we had structures for these two, and we were able to do what we thought was a really high quality alignment of these regions that we knew to be most important in the new-like subgroup. And so I think this is a strong alignment. And the fact that you see multiple proteins through those subgroups that have conserved elements within a subgroup that are different from the few conserved elements across the subgroup, like this little motif everybody has, that's really important information to be able to think about. And again, this is a starting point. We typically, I would say, have very shallow understanding of individual proteins in the superfamilies we work on, because we're trying to work on the whole set and multiple superfamilies. So we inevitably team up with experimental experts who have deep expertise in their particular area, because we just don't have the insight to then take those questions and ask the next step in a really, really effective way. A different example, here's the, I'm going to tell you a little bit about the origin of substrate specificity in a very small part of the alkaline phosphatase superfamily. Here again we have about 4,500 representative nodes representing 14,000 sequences, and we're interested in some of these NPP proteins. You can see these are the alkaline phosphatases, NPPs, pentomutases, etc. These are the sulfatases. They form 2 very different groups, which do indeed track with what we know about differences in those very distinct proteins. If we look at the oxotaxins, which are in this particular node, representative node, it's a specialized NPP. that recognizes specific ester substrates. When you look at the sort of canonical representative NPP, this is what the nucleotide binding pocket looks like, surrounded by the important active site residues. We already knew that one of the differences between the NPPs and the sort of canonical guys was that autotaxin is missing a structural element, which was thought to be associated with it being having a bigger substrate. When we looked at the network, we could see that it wasn't just one protein. There's a whole class of these. And the group is shown right here. And over here, you see the regular NPP with a red loop. The NPP, the loop that is present in NPPs that's missing in the autotaxin. So again, it's a way to kind of move from a bigger scale view down to more detail, a different kind of project, but still related to the theme I'm talking about. This is a slide that we did many years ago that we haven't done an awful lot of this, but I think it's really instructive to give you an idea of the different kinds of things you can do with this approach. So here we were working with a group interested in drug targets in parasites. And we did a very, very simple experiment. We took 7 sort of, and this was just a kind of model system experiment. We took 7 known human kinases and blasted them against the human proteome so that we could see the relative representation of these different kinases. Does that make sense to everybody? We then took the same kinases and blasted them against Giardia genomes, or what we had of the genomic information at that time. And what you see is that while the NEC1 kinases are very poorly represented in humans, they represent the majority of kinases in parasites. And so that gives a kind of platform for starting to think about where might you try to think about drug targets where you would have less cross-reactivity. Again, this is just a model. It's sort of a toy problem that we just thought we'd like to see what we could learn from this. So this is a very recent collaboration that we're doing with Chris Whitman that I think illustrates some of the more interesting findings that we're getting from the application of the networks. The 4-oxalocrotonate tautomerase superfamily now has 5 known main reactions that are rather different from each other, and 2 of them are shown here, the cis-CAD reaction and the 4-OT reaction. And in green is the 4-OT structure, and in this sort of brownish orange is the 4-OT, and the cis-CAD is down here. And you can see that structurally overall, they're quite similar. There's a fairly complicated domain structure in that the canonical 4-OTs that have been characterized are a single chain that requires both an A and a B chain to form a dimer, which then forms a physiological hexamer. In most of the other members of the superfamily, there are 2 fused chains, so they're twice as long. And what's key to this is that an N-terminal proline is the catalytically required element for catalysis in the 4-OTs, which have been studied extensively. The others have been studied less. Some of these fused guys in different subgroups of the superfamily have a proline in the middle that would sort of be the beginning of another chain if it weren't fused, but it's not a free proline, and you need a free proline at the N-terminal to be catalytic. So we haven't sorted out the complexities with that issue, but what we have done is we built a network of the whole superfamily. This is the group that is the cis-CAD-like subgroup. We're pretty certain at this point that this is a very specialized protein, and we don't think most of these do this reaction. We have no idea what the other substrates might be. But this one has been characterized. And this is the canonical 4-OT that Chris characterized a long time ago. And there are a few other proteins that we know in this group that do the 4-OT reaction. But it turns out that while you could see there are many different sort of emergent clusters here, we think there are multiple reactions in this group that we're just now starting to take apart and build full networks of the smaller sets to start looking at the differences, which I'm not going to talk to you about today. So what we wanted to ask is, what is different between the cis-CADs and the 4-OTs, and can we use that to understand something about how evolution produced sort of transitions that resulted in the two different reactions? So the evolutionary question is tricky because we only can work on extant proteins. We don't have the ancestors. But when we find the best links between two proteins that have different functions, and we can fill in that space with current extant proteins, we sometimes can start to see how they differ from each other along that transition. And it looks a little like a speciation plot. And it gives us some insight into the kinds of experiments we might want to do to try to engineer one reaction from another, or to at least explore that space and think about engineering new reactions, for example. So what we did is we took this region here and asked, what did those linkers look like? And here's a blow-up of that region, and this is the canonical 4-OT. Turns out that was there a question? No. It turns out that there was a fused 4-OT in this group that does have the tautomerase activity. And so here's a structure-guided alignment. So we took structures and superimposed all of those and built this alignment from that. So it's the best quality alignment we can get, given that these proteins are very diverse from each other. So we have the 4-OT, sort of the fused 4-OT is the next one here, and then we have what we call linker 2, which is the next linker that would be on a track toward the cis-CAD protein. Linker 1, which is closest to the cis-CADs. This protein that is doesn't do cis-CAD activity, but we have a structure, and so it was important and because it helped to link to the cis-CAD. And now you can see in that alignment, they all have the N-terminal proline, but you could start to see some sort of two different groups emerge until you get to the cis-CAD. Cis-CAD itself has some additional residues that appear to be important for activity and that are conserved in at least some of the members of this subgroup. And so now we have the beginnings of an idea of what the differences might really be about. And again, this is the value of the larger context. If you just compare one cis-CAD that's been experimentally characterized to one 4-OT, you don't know very much about all the other proteins that might give you some insight into what those differences might be. We actually, yes?
How comfortable are you when there's only two?
We actually have an alignment of 200 sequences, and these patterns hold.
Oh, okay.
Yeah.
So there's more Y, let's say more Ys
Yes.
in your long.
Right. In fact, almost all of these guys have a tyrosine in that position. There's a tryptophan in some of these. Well, I don't see it, but there's a tryptophan in some of the 4-OT groupings, and when we broke those down into smaller groups, we also saw big blocks of conserved residues. I just couldn't show an alignment like that, but good question. Okay. There's a preliminary kinetic analysis shows that as you move from these up to these, you do see a loss of the 4OT activity. They haven't seen activity for CISCAD. We think, we really don't know what the functions are of all of these proteins. And since this is a specialized sort of outlier biodegradation type protein, there could be many different substrates and reactions along that way. So testing that, we may have to, what Birjan, who's the postdoc in Chris's lab, is doing, is trying to make mutations in the CISCAD to make them look more and more like 4OT, so we can see if that's sort of a sensible way to think about this. The other thing that we're planning to do that we haven't done yet is to add data from metagenomic projects in so that we have a bigger sequence set and can do a better job of asking, what's allowed at those positions for groups in these different groups? Are there multiple other subgroups that might be linkers? It's just the start of this project. Nobody's talking or asking many questions. You're welcome to. Yes?
I could see an opportunity for directed evolution to sort of help you.
Absolutely. Yeah, and Danny Tofik is doing some things like this. So you take your whole set, you use those as sort of basis variations, and then you do a directed variation around some of these different motifs. And then if you want a selection, if possible, if you could pull out some winners, then you'd be able to really take this apart experimentally that sort of matches the big computational picture. And I think that's a very exciting direction for this. We can also do things like on our networks, we can map active site residues and ask, are they found in all of a particular group? Maybe if the representative that you've been working on is rare and it's not really typical of the whole set, that's really important information, and that's why that bigger context is useful. One of the reasons. We can also do linkers, find linkers in just computationally in an automated way, and this is an example from our thyroidoxin project from a long time ago. And that was at one point, Shelley Copley and I were looking at peroxyredoxins and thyroidoxins to try to get a handle on how they might be related, and if thyroidoxin was kind of an ancestral type for the development of, for the evolution of peroxyredoxins. And so it took us 6 months of effort. And the way we did it was we used a little program that we had developed probably 20 years ago called Shotgun. And what we did was we took a bunch of peroxyredoxins that were less than 30% identical that kind of sampled what we now know to be this space. We didn't know that at the time. And a bunch of thyroidoxins, and we used those as queries to blast against the database. Then we rank ordered the hits by how many of those queries found that hit, which is essentially what we're doing with the network, so only on a much larger frame and more elegant way to do it. And what that showed was that there was a particular protein called, this is a co-migratory protein in the thyroidoxin class. It has a specialized substrate, but it does the thyroidoxin reaction. And we found that the CMP could link the thyroidoxin and peroxyredoxins. We didn't have all these structures. All these big nodes are structures now. So this was the paper was published in 2004. But we could show that going from CMP to the different types of peroxyredoxins, you could read off the active sites, the change in the active site, the move of the active site cysteine and the cis-XX cis motif to the second cysteine, the change in the cis-pro. You could practically read off of it just going from CMP through the different peroxyredoxin types. And this just shows the ornamentation of the various peroxyredoxins as you go through those. So that project took us at least 6 months of very hard work by quite a few people. This took no time at all, essentially, to ask the question, which you can ask of Cytoscape, what is the closest path between, say, this protein and this protein? And when we did that, this was the path we got. Now, this was found by that analysis, and that protein wasn't in the database when we did this work. So you can see there are a lot of ways to try to get at these kinds of linker transitions and to give you a toehold on next steps.
So Pat,
Yeah.
a lot of this available to sort of the general public via
This is in Cytoscape.
Right, but so they can generate the networks via the web-based type stuff?
Yes.
Okay.
And then that's one of the things, that's a Cytoscape tool. And Cytoscape has gotten much more expanded over the last few years. It has an app store, and there are, probably a couple hundred apps in there for various things you can plug. They're basically plugins that you can do all kinds of analyses. Now, most of Cytoscape is directed toward interactions, whether they're protein-protein or genetic interactions. That's right.
That's what I'm talking about.
That's right.
So I'm sure you guys just have, program sort of routines that you run through.
Right.
For the rest of us who aren't computational.
The EFI server, you can use that.
Oh, you have a server? Oh, okay.
No, the EFI does, the Enzyme Function Initiative server. And I gave the link on here. Yes. The reason we can't do it on a large scale is that we generate not only the superfamily network, but then we generate all the subgroups and smaller, different levels, including full networks, as part of a curation for specific superfamilies. And it used to take, until very recently, it used to still take us weeks to generate some of these networks. Now we can generate them much faster, but I'm really a biologist, and my lab doesn't have the resources to do this on a really large scale. So we simply have to do it collaboratively on specific projects. But you could generate networks there are a bunch of servers out there. A bunch of them don't work very well. The EFI one does give you networks, and they'll look somewhat different from ours because they generate the networks a little bit differently. And I'm going to talk about that a little bit more at the end because there are lots of challenges and caveats. And
well, Patsy, can I ask a question? So you generate a network, all right? You put it in Cytoscape, you can visualize it, and then you take certain clusters, right? And can you take a cluster and put it back into the EFI server?
yes,
Oh, you can?
I think you can. Yeah.
Okay.
Yeah.
I've
So
never done it through the server. John is giving me access to it.
right, that's right.
The mainframe computer.
Right.
Everybody else.
Yeah, for people interested in Radical SAM, we have generated all those networks. And you could just download them from our database.
So we can take the methyltransferases, right, and then just...
That's right. And Gemma's made very detailed curation that's been updated recently. We're still trying to get that paper out, but we keep having to work. It's a really big project. So yeah, I think it has to be accessible to other people. One of the things I hope to leave you with today at the end of the talk is some of the things you have to think about that are just technical issues about challenges for interpreting these and how to use them. So, any other questions right now? Yes?
So when you draw those lines, what are you thinking, like, under the hood? Are you thinking, the shortest the distance, or you're looking at the clusters, like, saying from the cluster, because that might be more accurate, or, more robust, because, what we see here is not ancestral.
Very, very good point.
It's what we see.
Yes, very good point. And I am going to address it a little later in the talk. But the short answer is you could use clustering algorithms like Markov chain clustering, which we've done with the GSTs, for example, And it will break your network up into clusters that are pretty good. And I say pretty good based on what we know about the biological bins. They're very difficult to parameterize for that inflation parameter for a given superfamily because they're very different from each other. So, and that's why we use thresholds, is so you can explore the space, the whole space yourself at different levels of similarity. Now, the layouts are also different, and I'm going to talk about that. The layouts I'm showing you now track pretty well with distance, but it's a proprietary layout, so we don't know exactly how it works. So I'm going to show you another layout that is basically uses spring constants. So they correlate directly with this dissimilarity.
So the distance between nodes here actually
is the order of connectivity. But it turns out that's, if they're highly connected, they're highly similar. And it does indeed. What my student did at the time is took the networks and counted the pixels, because it was a proprietary, so we didn't know how it was built, and simply counted the pixels in all the edges and asked how well they tracked with similarity. So, but it's a good, and I will talk a little bit about it more in a minute. Okay, just moving on to sort of expand the picture a little bit. This is from a mini review that Shoshana and I published, I guess 5 years ago. What we did is we took a subgroup from the inlay superfamily, and all the colors represent known functions. And we used the human gut microbiome because it was well curated and we had the protein sequences, and we just simply asked what are the sequences that map to this subgroup, and they're all shown in brown. And if we change the threshold to be more distinct so you can see this better, you start to see some patterns. Like here, I think these are the galactonate dehydratases. These are the fuconate dehydratases. And there are some brown nodes in here that immediately suggest, because these separate very nicely in terms of, they separate very nicely in terms of the different functional classes in these acid sugar dehydratases. You know what the function is. You have a very good hypothesis for the function of that function's present in the gut microbiome. What's especially interesting is everything in white is a protein in GenBank at the time that had an unknown function. And so you see a bunch of unknowns that just are there, but you also see some clusters from the gut microbiome that don't look like anything in GenBank or anything that we know about. So there are probably some new, probably acid sugar dehydratases, because all the proteins in this subgroup that we've found so far, with the exception of mandylate racemase, are acid sugar dehydratases. So this is a way to fill out your picture in a very powerful way, because it adds a lot of data. Again, if we actually are thinking about it as a way to help annotate functions of metagenomic sequences. But I think this is really promising to even, I think the bigger the context, the better for certain kinds of studies. Fast forward, you can now do this on a very large scale. So this is a recent analysis of a different set of proteins. This is actually from the nitroreductase superfamily, where there's a server I gave you the reference here, that has about 1,800 metagenomic projects where the proteins have been called. Now, those are not going to be great in terms of the best calls of the proteins or full length, but it's a huge amount of data. And what you can do is we took our hidden Markov models and the one we had for this subgroup and ran it against this whole set. and found where proteins that looked like this set of proteins were enriched in these different metagenomic samples. So then we could color them whether they came from soil or sludge or water or one of the gut studies. So now you could start saying, gee, I really care about ruminant guts versus human gut and what's the differences, or I'm looking for particular kinds of functions that I might find in one environment but not another. And you could start to take apart these very large data and map it to your networks. And again, this is just kind of another way to think about doing this kind of analysis. I'm talking about a hammer looking for a nail, but that was the intent in this talk, was to just give you a smorgasbord of some of the ways you can think about this. Okay. Some challenges. I think it's really important to talk about Ds. And I hope there'll be some discussion either now or later. I'm happy to talk to people about any of this offline. We've been worried for quite a long time about the enormous heterogeneity in what's actually being picked up when you look at a two-dimensional representation of the networks. And there's just a lot of human assumptions that go into interpreting them because they are so intuitively accessible. It's really easy to make inferences that can't be, that you can't really trust. And so the first thing I would say is you've really got to do something more. It's just a starting point. Again, I see it as a hypothesis machine and a very powerful one, but you've got to, like any experiment you would do, you've got to step in and do the hard work and the detail, and you've got to use your controls, or you're probably going to get garbage out. So I want to talk about that a little bit because I think it's important as scientists to actually be able to get your hands around this in a really useful way. So there are a lot of technical challenges that have to do with the speed of generating the networks, their size, being able to visualize. Since you can't visualize a network with more than about 500,000 edges, and we have like 1 billion edges in the full network of the radical SAM superfamily, you have to use very compressed representations that lose a lot of information. And it's still very useful to build the whole radical SAM network and take it apart into major subgroups, ask whether they map to the very complex domain structures, and then tease apart one of those groups at a time to start working on the project you care about. That's really useful, but you really have to use it as a rough guide because it's no more than that. The signal that gets captured, you have n-1 pairwise comparisons when you do these all-by-all comparisons. And that means you're generating a network that is at high dimensionality, where n is in the tens of thousands or 100,000. And so you're smashing that down into 2 dimensions to look at the network. Now, we've shown that you get an awful lot of the information out of that. But depending on the system, you can infer similarities between 2 groups that are very, very diverse because they look close together on the network that aren't real. So again, you've got to drill down and ask the question more thoughtfully. How to choose and automate appropriate thresholds, that's really a hard part. You can give up and use clustering algorithms, but they're not very helpful in summarizing large-scale relationships because the way clustering algorithms work is they optimize the closeness of each clique. So you're getting lots of smaller groups that hang together pretty well, but what we want to know is how large group A relates to large group B, C, D, and E. And so the thresholded networks are very useful for that kind of exploration. And we're just starting to understand how to choose the thresholds we want for an appropriate question is actually a manual effort that takes a lot of thinking. And I would say that the people in my lab and a few others who've thought about this now for years, because we've been doing this for 10 years, and we're starting to get our heads around how to think about some of the questions. But it's you kind of have to spend quite a bit of time with it to be able to do that.
So Patrick, I have a question.
Yeah.
So are we going to move into sort of network by network networks? In other words, so take radical SAM, for example, where one of the big issues is what are the substrates, right? So can you cross a radical SAM network or a particular cluster within a radical SAM network with sort of hundreds of ligand binding networks?
Yeah, there's no reason you can't do that. The code, there's no tool to do that right now.
Like say, the easy one would be, tRNA binding proteins.
Yeah.
Right? And do, radical SAM cross tRNA binding proteins and see if they intersect.
Yes, and right now, we don't have code to automatically say, I want to take this cluster and this cluster that are just there in the big network and compare them directly, unless you make a new network just of those sets. But when you do that, you may have intervening groups that you didn't know about. This happened with the alkaline phosphatases, where it has been thought for years that the alkaline phosphatases and the NPPs were each other's closest relatives because they're mechanistically the most similar. And you just have that third metal in the alkaline phosphatases. It turns out the mutases connect them best. And if you want to align them, you want those mutases in there. And so I think we ultimately need a suite of tools that will really do this. It's going to have to be somebody else than our group, though. We're not really principally computer scientists or software developers. And I think the Cytoscape development team might do some of this. They're starting to get protein similarity and similarity networks on their radar. But by far their biggest business is interactions. So I think we're still very early. It's just, I think we're pretty sophisticated given that we've only started thinking about doing this about 10 years ago. But we're still just really babies in terms of getting to where we would like to be. So interpretation of the networks is really sensitive to lots of things. If you remember the RepNet and the full network of that new class GSTs, those networks are pretty different from each other. And you have to know what you're looking at and how the data is compressed, not in detail, but it's best probably if you could make a RepNet and a full net of some group you care about, because then you can use both approaches. And I think the name of the game is pull in as many different ways of looking at the results as you can, because you're going to get a much richer and more thoughtful interpretation out of it. Complex domain architecture. Radical SAM has been killing us. If you look at if you look at the network, and you could take for the PFAM domain, you could make an ARC schema picture of all the domain organization, and it's this huge, very complicated network. Well, every time you have multiple domains, and you're doing say like you have a spasm domain and others, but you add spasm to the sort of radical SAM binding domain itself, all of those proteins are going to be clustered more closely together than they are to the proteins that don't have a spasm domain, because they have more connections and the connection's longer. And if the spasm domain has higher similarity within it than the radical SAM binding domain, that connectivity is going to be dominated by the spasm domain. So the way we're kind of moving toward is saying, okay, let's make a big network of both just the radical SAM binding domain and of the full-length proteins, and ask, is the topology very different? And we are putting that in our paper. It's actually pretty similar, but it's important to look at it. And then we divide and conquer. We say, okay, let's take all the proteins with a spasm domain and treat them as a separate network. And now we maybe want to drill down to level 2 or 3 level subgroups to take it apart even more.
Do you see proteins with spasm domains that aren't about the same?
You know, I don't remember the answer to that. I think there may be some spasm domains out there, but Gemma would certainly know, but I just don't. Yeah, you should ask her. The other thing is that predicting functional boundaries is really, really hard. And that's why, except in trivial cases, it's hard to predict functions correctly. You get uneven rates of evolution in a superfamily. What that means is that the OSBSs in the enolase superfamily evolve very much faster than the other families. So most of the OSBSs across a given group of organisms are less than 20% identical, whereas the enolases may be 60% identical. So when you look at the network, they're going to look like the OSBSs are going to be a whole bunch of separate clusters, and the enolases are going to cluster tightly. And so you might think all those different OSBS clusters are different functions, and the enolase cluster is, the enolase family cluster is one function. And until you get into the biology and sort of start understanding that, you're not going to be able to interpret it correctly. Don't let the prettiness of PSNs fool you. I was talking to somebody a few years ago whose wife had a business making art from scientific images. It was somebody like Wolfenden's wife, or somebody like that, and wanted to make some pictures out of networks because they're really pretty. Some of them are quite beautiful, and they're high resolution. And you go, oh, isn't that great? So don't let it fool you just because they're pretty pictures. You all know this, but it's really compelling when you see the pictures sometimes to think some interpretation that may be unfounded. So I mentioned a couple of these, and I'll let you look at those yourself because I want to show you the pictures. So here's 2 different layouts. This is the organic layout that I've been using in my slides, mostly because you could show a large network as a single image on one slide. If you use a force-directed layout, we're using spring constants that capture the correlate directly with the lengths correlate directly with the similarity. The same picture would be across this whole wall. It's very hard to see it. So this is actually a great layout to work with and use as an example. Here's another variation called perfuse force-directed. So you are using a spring constant to capture the similarities, and length is correlating with the constant. You can see some differences. It's also been, essentially, it's been, a constant has been added in there to minimize, to diminish quite how expanded the picture is. But there are other layouts that look quite different. And depending on how you parameterize this one, you could get networks that look really very different from each other. Oh, I didn't remember. So this is the domain organization network for radical SAM. You can see it's really, really complicated. And so even looking at the guys that have a spasm domain, they're pretty complicated too. And so you can't just say, oh, good, there's a spasm domain, that's what it looks like, that's how big it is, that's what it is most similar to. From this network, you've got to look at a more granular network to really help you ascertain where it kind of falls relative, say, to the biotin synthase-like guys. Alkaline phosphatase doesn't have separate domains that can be easily defined. Instead, they have big inserts attached to the Rossmann fold. There could be multiple insertion places, so it's a complex picture. And the problem here, we wanted to take this apart and just use the Rossmann domain only, which we were able to do, but it was a huge amount of work to figure out what the boundaries were between these domains across the whole superfamily, because nobody had done that. So it had to be done carefully before we could start to make that domain-only network. And so that's another kind of complication you run into. Kind of to wrap up this section, and then I have just a little tiny bit about genome context networks. I've said this many times, and I hope I said it enough times to convince you that it's really just a starting point. One of the things we've learned about studying about 20 of these big superfamilies in some detail is that each has evolved in unique and complex ways, whether it's variation in domain structure or where the active site is or how the structure has varied to deliver function. What are the sort of fundamental architectural differences that could be associated with evolution of functional specificity, so that you can't build simple generalizations. I can't say, well, if you use a threshold of 1 times 10 to the minus 25 for pretty much most superfamilies, you'll get reasonable functional boundaries at a gross level. It's not true. So most of my friends in the computational community are always moving toward a generalizable approach and a set of principles that are general across a whole set. And at the level at which biologists and chemists need to look at these proteins, it's my opinion that you just can't do that. There is no threshold that works. There isn't even a threshold that works for all the proteins in a given fairly simple superfamily. If I want to cluster the OSBSs together in the enolase superfamily, everything else is a hairball. So I have to break apart that OSBS family and then look at it at a different threshold to be able to sort out those relationships. So that's a really complicating issue. Again, the more orthogonal and rigorous approaches really have to come into play to evaluate your networks. I talked about this. And the other thing that is both astounding and difficult for the field is that so few proteins have been experimentally characterized or looked at in any way, even with like high throughput proteomic studies. And that means you don't have very many functional anchors to make an inference about unknowns. And so it becomes the problem appears more and more difficult as time goes on instead of less, and I think that appearance is real. So I'm going to talk about a tiny bit about genome context networks. It's a different animal from protein similarity networks, although it's related. So there's a lot of interest in these networks because we already know that in bacterial systems that there are some pathways that are co-localized on a genome. And for example, especially in plasmids that travel around and do what we think of as biodegradation, they're just looking for a new ecological niche. That will happen frequently. For E. coli and Subtilis, we know quite a lot about what those models look like. For the huge proportion of other bacterial species, and I'm not a microbiologist, we don't even really know what those models look like and how far apart the genes are, what intervening genes there might be in a pathway. If you're lucky, you find a regulon that's co-regulated and co-localized. And it's a powerful way. If you have an unknown and you can place it in a pathway, that's a really great clue for function. And if you put that together with things like I've been talking about today, you might get a good enough clue to be worth a graduate student spending 5 years trying to run that down. And I mean, that's what you're getting down to. It's a very big time and intellectual effort overhead to try to figure out a function like this. What we do is we take a set of query proteins that are related to each other. So we could get genome context for the whole set. Because oftentimes when you look for genome context, I don't know how many of you have done this, you kind of don't find anything useful. You might find 2 proteins that co-localize, and one is a dehydrogenase, but that doesn't tell you much often because you don't know anything more about it than that. And so sometimes you find a whole pathway. The mandylate racemase pathway, the beta-keto-adipate pathway is great. It all co-localizes. It's a central clearinghouse pathway for aromatic catabolism. They're just lined up nicely, well understood. It doesn't happen very often. You have a better chance if you have multiple shots on goal, and that's what we're trying to do, where instead of taking one protein and trying to find its genome context in many organisms, we take a bunch of related proteins and try to find all of their genome contexts in many organisms. So as you can imagine, this is a big computation. So what you do is you take your set of proteins and you find all the genes and the proteins upstream and downstream from them that could be co-localized. And then you generate networks from those where each one of these is represents the genome context for one of those proteins. And then you can color these. Let's say these come from 2 different phyla or 2 different types of bacteria. And you can color these neighbors by the queries they map to. So here you've got a little group where all the queries came from here. So you know there might be some consistency across the set. And you may not have found for all these red guys a really great signal, but if you have multiple red guys or multiple green guys, you have a little bit better signal. And here's a real example that Shoshana did where she took, these were query proteins, and this happens to be from gene B, and all of these clusters are homologous proteins that are homologous to something in the query set. So you've got lots of homologs. So you're doing this in multiple dimensions. And then she was able to find genome context answers for several of these different genes. And she's colored them here by the number of hops they are from the query sequence. So typically if we take 10 genes upstream and downstream, or 5 upstream and downstream, the strongest signal is going to be close and not too many hops away. When you start to get more complicated, you might have 2 steps in a pathway 4 genes apart with stuff intervening that isn't relevant, but often you're basically looking for the low-hanging fruit. But the problem here is this is a very, very large computation, and it gives you lots and lots of these. And sometimes the ones that are the most important only have a few hits because they exist only in species that live in a certain kind of weird environment. So they're easy to miss. So these are really complicated to interpret. But if you get a great big cluster and there's a bunch of homologs for each one of these proteins, then you start to, you've got a strong signal here. And this happens to be the context network for a well-known pathway. So just to give you a little bit of a hint about the kinds of things you can do, again, with a large-scale context. So I finished exactly on time. I acknowledge a lot of people who have been in my lab, are in my lab, a number of collaborators over the years, not everyone, of course, but I hope this was useful to give you sort of a way to start thinking about these. And I don't know, do we have time for questions?
So Patsy, for the genome big networks, are the most hits the ones that are in clear operons, or do you see some that are, for example, transcribed in a different direction, but are within 10?
Yeah, you'll see both. You often want to look for the biggest ones because then you have a kind of consistent signal.
Yeah, but are the biggest ones, I guess, the ones that are usually in an operon, co-transcribed?
They could be, but you often will see a couple of them are in an operon, and the others in the operon might not be. You might not see a big set for whatever reason. Yes?
So maybe you could just briefly touch on, I've typically seen the hidden Markov state model used for better exploring base computational space or for extending timescales of molecular simulations. How are you applying it here?
Here what we do is we make a model for a set of sequences that are homologous that we've aligned. And so we make a profile that takes all that alignment, and for each position in the alignment, vertical position, you're looking at the probability of finding whatever you see from your training set in that position.
So each position is a training set?
Yes. And so choosing a good training set is important. We often find we cannot get good models for whole superfamilies because they're so varied. One of my senior scientists has developed some code to take an HMM and find the sequence that best represents the profile if we want to do something with an individual sequence. And we've started to do similarity networks with HMMs, and they're quite similar in topology to the blast-based networks, but they're a more rigorous approach to do it. It takes a lot more time. Yes? We make the assumption in all of our networks that the proteins are homologous. And because we're assuming that that similarity comes from that homology. Now, if you were going to take two sets of proteins that were convergently evolved, it might be that you might pick up, the catalytic triad, for example. And that might be the only part of the signal that you're picking up, and that could join those two groups that would not have to be homologous. And because, that's just sort of an underlying assumption, but you could certainly do that. And I think in radical SAM, we are not sure, maybe other people are, but we are not sure that all the proteins we include in the radical SAM superfamily even come from the same fold class that are homologous. Because at the level of the whole superfamily, you're mostly just picking up the SAM binding motifs. And that motif seems to have been used in lots of different proteins. And if you superimpose structures around that motif, you get a good superposition. But the rest of the structures don't look that similar. And we asked Andre Schally to thread the sequence set we had onto structures from his database to see, because we want a little more feel that they really were homologous. And they couldn't really call the fold class of all of them. Now, that's just these are so diverse it's really hard to say whether that's meaningful. We don't worry too much anymore about getting the most diverse set of proteins possible because the tools are very good, but when you get beyond a certain level of diversity and you're just using something like BLAST, or even when you use structure-based, so you can see a lot further with structure, even there you may be only picking up a small part of the active site. So I think you're getting into very, very gray space there, and you have to know what you're actually put in so you can interpret better what you get out. Did that answer your question? Okay. Thank you.